Good evening. My name is Paul Nurse. I'm president of Rockefeller University, and it is, of course, a great pleasure to welcome you all here to the campus and to this auditorium this evening. Now, in a minute, I'm going to tell you rather more about this evening's program. But first, I'm up here to introduce to you Jeff Smith, who is chair of the Parents and Science Initiative, who have organized this evening's program. Jeff first became involved with Rockefeller in 2006 through the university's Bridges to Better Medicine program and joined its steering committee soon thereafter. He's also a member of the Rockefeller University Council and in addition is a visiting scholar here at the university and runs a very successful, and indeed is the founder of, Science and Economics program, offering courses which are very popular with our graduate students and our postdoctoral fellows. So Jeff's involved with many facets of our campus life, and so it's a great pleasure to have him here. Jeff, could you come to the stage?
Thank you, Paul. It's a pleasure to welcome you all to our Spring Parents and Science Evening. This is the third and final event that Rockefeller University is hosting, especially for parents, during this academic year. The university introduced Parents and Science lecture series last year as a resource for New York City parents interested in gaining new insight into science in general and child and adolescent development in particular. The Parents and Science programs also seek to provide parents with time to see each other in a thought-provoking environment. And as I'm sure most of you would agree, this is time that's often hard to come by in the busy schedule of average New York City parents. We have been very delighted by the enthusiastic response that Parents and Science has received. More than 300 people, for example, RSVP'd to this evening's program, and I'd like to thank you for joining us tonight and hope that you all become regular participants in the dialogue that we're hoping to foster through these programs. I would also like to take this opportunity to thank the trustees of the Lubin Family Foundation, some of whom I believe are here this evening, who have generously underwritten the second year of the Parents and Science Initiative, and their ongoing support for this series is greatly appreciated. Before I turn the podium back over to Paul, I'm going to take this opportunity to tell you a bit more about him, as he tends to often be the introducer at these events rather than the introducee. I'm going to take this opportunity to spend a few minutes. Paul moved to New York in 2003 to become the head of Rockefeller University, and during that time, or since then, he's become one of the nation's most prominent voices in science. And in fact, when Paul first arrived on campus, he stated that he wanted to make public outreach and education about science a top priority. And I think he is certainly achieving this goal. A number of prominent publications, including the New York Times, the New Yorker, and many others, frequently turn to Paul for his comment on important issues in science. You may also have seen Paul on The Charlie Rose Show, where for 2 years he served as the co-host for a series of episodes reviewing various issues related to science and health. Paul, of course, is an extremely distinguished scientist, and the list of rewards that he's received is too lengthy to go into detail, but there are a few recognitions that certainly stand out. In 1998, Paul was awarded the Lasker Award for Basic Medical Research. This is an award that is often referred to as the American Nobel because it often presages receiving that award later. In 1999, Paul received a knighthood for his contributions to cancer research and cell biology. And in 2001, he did in fact share the Nobel Prize in Medicine for research that shed light on cellular mechanisms involved in cancer. So with that, it's my great pleasure to welcome Paul back to the podium.
Jeff's also a very nice man, I have to say. Well, I hope tonight's program will better acquaint you with this quite remarkable institution that you're sitting in this evening. It was the first of its kind as a biomedical research institute when it was founded by David Rockefeller's grandfather in 1901. And since then it has made many, in fact very many, important contributions to biology, biomedicine and medicine recognized the world over. I will tell you of one of them, and that was the discovery that DNA is the basic material of heredity. That was discovered on this campus, published in 1944. Not a well-known fact. We all tend to know about the structure of DNA and Watson and Crick, but actually the fact that it was the basic material of heredity which set the foundations for the molecular biology and molecular genetic revolution that we're now still in the middle of was in fact discovered here at Rockefeller. 23 scientists have become, who are associated with Rockefeller, have received the Nobel Prize. This is really quite an extraordinary track record of success. I want to put that number in perspective. If Rockefeller were a country, rather than a very small research institution, it would rank 4th in the world in Nobel Prizes in medicine and chemistry, behind only the United States, Britain and Germany, and just ahead of France, that would rank 5th. And that, I should say, is with a faculty of no more at any one time than 70. No more than 70. Harvard has about 5,000, if you want to make a comparison. Not that we compare ourselves with Harvard very often, actually. 4 of Rockefeller's 23 Nobel laureates received their prizes in the past decade. But there are other metrics of the university's excellence, because there is some luck associated with these big prizes, I have to say. 20 Rockefeller scientists have received the Lasker Award, 13 the National Medal of Science, which is the nation's greatest scientific honour. But what I find actually the most important metric for me personally is well over half of our current tenured faculty are members of the National Academy of Sciences. And why I mention that is that we have quality in depth in the majority of our faculty. Refer to your programs if you want to learn more about Rockefeller and the university today. You might also want to look at our new, it was only put up a few months ago, a Nobel and Lasker Award wall, which is just out of the lecture theatre on the left as you exit the auditorium. Now, in your program booklets, you will find an editorial from yesterday's Daily News. This is an editorial that took us completely by surprise, I have to say, and it's entitled Geniuses at Work. New York City's Rockefeller University is the nation's brain trust. And one of the brains referred to in that article is, in fact, our speaker this evening, Dr. Jeffrey Friedman. Mentioned 3 people there in that article, and in the editorial, and one of them was Jeff. Jeff's topic tonight, up here, is Science 101 for Parents, or 20 Years of the Genetic Revolution, in 1 hour, perhaps a bit less than 1 hour. We have to talk to him about that, but certainly in a short time. Jeff received a medical degree from Albany Medical College at the age of 22. This is quite young. He went on to earn a PhD in 1986 here at Rockefeller and liked it so much that he couldn't leave, and he's still here with us. And we're all very grateful for that, I have to say. Today, he's the university's Marilyn M. Simpson Professor. He's head of the Laboratory of Molecular Genetics, director of the Starr Center for Human Genetics here, and is also an investigator with the Howard Hughes Medical Institute. This is a highly prestigious position. There are about 300 such investigators throughout the country. We have 13 or 14 here in our faculty, and Jeff is one of them. His research focuses on the genes that regulate appetite, body weight, and energy metabolism, including genes, of course, associated with obesity and diabetes, and is noted for his discovery of the hormone, which many of you will be familiar with, which he named leptin, a major breakthrough, the discovery of this hormone, and its understanding in the mechanisms that control eating behavior and body weight. And for this work, Jeff has received many honors, including a Gairdner Foundation Award, which is a sort of precursor to a Lasker and Nobel, and also, of course, election to the National Academy of Sciences. After his talk, we'll take questions from the audience, so please hold your questions until that time. So I'm delighted to ask Jeff to come and join me here at the podium.
Thank you all for coming. It's nice to have the chance to tell you about some of the recent developments in DNA research and how it might be relevant to our future and, more importantly, our children's futures. Now, I was given the following title. I missed the second half of it that Paul read to you and that is in your program. And you might notice this is a rather general title. I wasn't really sure what to do with it. I knew that I was being called on to talk about genomics, and so I began to wrestle with some possible titles, the first of which is shown here. 20 years of the genetic revolution, 130 years of genetics, the movie Gattaca, healthcare for our children with some assorted generalities, and an opaque subtext crammed into 1 hour. This would have been fine until I found out that I only have 45 minutes. So I tried to come up with a more succinct title, and that is here. How come I look like my parents and not a giraffe? This seemed a little banal, so I considered a third title, which is Unraveling Our DNA. Now the fact is, I'm going to try to cover all of this, and if I'm successful at that, you'll learn, hopefully, or more, hear about some of these things. If I'm not successful, of course, this is an hour of your life you'll never get back. So Paul mentioned the seminal discovery here that DNA was the genetic material. I'd like to begin by mentioning that yet again and telling you about the people who did this. Their names were Oswald Avery, Colin MacLeod, and Macklin McCarty. Many of us here, including myself, had the great privilege of getting to know Mac before he passed away a few years ago. And he was a fantastic person who could really bridge, for all of us, one of the greatest discoveries, if not the greatest of the 20th century in biology. Now, what these gentlemen discovered, as Paul mentioned, is that DNA was the genetic material. This was in 1944, and it generated tremendous excitement on campus, albeit with some controversy that I'll tell you more about, as well as the details of their experiment. But among the people who were excited about this development was a professor here named Rollin Hotchkiss, who in a flight of fancy wrote a poem which he called Revolution or Evolution. I'd like to read the first paragraph for you. Once a prehistoric blister in the prehistoric slime thought I'd like to have a sister and to save himself some time. He wrote down the mystic secrets of his protoplasmic virus on some stuff that he found handy, a sort of primitive papyrus. Now, what McCarty, Avery, and MacLeod had discovered was that the primitive papyrus was DNA, and that basic template, several billion years ago, followed by millennia upon millennia of evolution, led to the emergence of the most advanced organism, the human. And now the capability exists, as we'll come to in a minute, to identify all of the DNA elements that are responsible for the characteristic features of these individuals, their spirituality, the differences in their height and weight, and so on. The vehicle for much of this will be the ability to sequence DNA. And if you search the web, it's not hard to find companies that will actually sequence all of your DNA for you. This is one such company, KnowMe. No, this is not an X-rated site. This is simply a site where you get to know yourself through your DNA sequence. Has anyone here had their DNA sequenced yet? Many people have. Well, we'll talk about it because it's something that, if not in your future, is certainly in your children's. So what I'd like to begin by doing is tell you a little more about how we got to this point, what it means to sequence your DNA. And so this is going to require that I try to summarize 130 years of genetics in about 17 slides, at least insofar as telling you what a gene is, what DNA is, and a little bit about how this works. Now, this story begins in Slovakia with the work of Gregor Mendel, whose name I'm sure many of you have heard about. He lived between 1822 and 1884, and it was he who defined some of the basic rules of heredity. Now, Mendel faced a conundrum in those days that every plant biologist or every farmer faced, and it had to do with the fact that seed salesmen could sell you seeds and tell you with great precision what type of plant that seed would grow up into with respect to a number of variable traits. So, for example, the seed salesman could give you a seed and say that is going to be, for example, a short plant. Uniformly, this seed will, as it were, breed true. And the way you know you can do this is with plants you can self-pollinate. So if you self-pollinate this plant forevermore, it will always be short. A second seed could be delivered to you, and he would tell you that this seed will always yield a large plant no matter what. It will breed true. But there was a third type of seed that he could give you that was quite curious and that no one could understand the basis for. This seed, when selfed, would give a large plant, but when it grew up, would give a large plant, but as you selfed it, it would give both large and small plants. And no one could figure out the basis for this. Some seeds are small, some seeds are large and always large. Other seeds are sometimes large and sometimes small. Well, Mendel had a mathematical background, and he did a number of analytical experiments. But one of the key experiments he did was simply to take one of these plants that will always breed true short, a seed that will always breed true short, and another plant that will always breed true long or large, and mate them. And now we got a long plant, but that long plant would give both short and long progeny. Well, after puzzling about this, undoubtedly, Mendel came up with the following suggestion, that there are two distinct elements conferring heredity for every trait. You get one from the sperm and one from the egg. And he reasoned that the short has two copies of short, and it will always be short. The long will have two copies of the long trait and will always be long. But this different type of long or large plant, will have one copy of each. And so the basic conclusion from this is that there are two characters, one inherited from your mother, one from your father, or one from the sperm and one from the egg for every trait. If they're uniform, then that trait will be propagated forevermore, but if you carry one of each and self, then these things will distribute independently, and so a fourth of the time you'll get two shorts, a fourth of the time you'll get two longs, and half of the time you'll get a long and short. And these are the plants that will always breed short, these will always breed long, and these will breed both. Now, what Mendel had essentially described is one of the basic rules of heredity, something we now take for granted, which is that we inherit some traits from our mother, some from our father, we're an amalgam of both, and that these traits are propagated from then on. Now, regrettably for Mendel, his work was largely ignored at the time, although among his last words were, "My day will come," and indeed it did. His work was rediscovered and repeated by several other groups at the early part of the 1900s. In 1889, perhaps knowing about Mendel's work, although it's not clear, the term pangen was provided and by Hugo de Vries. This refers to the element of heredity that confers either shortness or longness on this particular plant. Now, Mendel described many other traits that are inherited in a similar fashion, and as I alluded to a moment ago, this was followed up on by numerous groups in the early 1900s. So de Vries calls this unit of heredity, this biochemical entity, no one could say very much more about it at that point. This biochemical entity controls one hereditary characteristic, but the nature of that hereditary element was not known. In 1909, a Danish scientist, William O. Hansen, shortened this term pangen to gene, and that's what we know a gene as today. It's the smallest unit of heredity. But what the gene was unknown to Mendel, as it was for Darwin. All it was known is that traits are somehow passed on from generation to generation. An important advance was, however, provided with the realization that these genes are passed on from generation to generation on chromosomes. This is an electron micrograph of a chromosome, and the key insight here came from the work in 1902 and 1903 by a scientist named Walter Sutton, who realized that chromosomes are of a particular number. Shown here, there are 46 in humans, one half from our mother, one half from our father. But what he noted in another species, namely Ascaris, a worm, was that when that worm generates a gamete, or a sperm or an egg, the number of chromosomes is halved. And so chromosomes themselves appeared to behave similarly to the factors that Mendel had imputed in his work. By that I mean you have two copies of each in the adult organism, and when you make a sperm or an egg, there are half as many. And so chromosomes emerged based on this and subsequent work from Barbara McClintock, who won a Nobel Prize many years later, and others, that genes are carried on chromosomes. So the question then is, what's the biochemical nature of a chromosome? What is it about a chromosome that it allows it to transmit heredity from generation to generation? Well, if you're interested in any biochemical question, and you think you've really fundamental one at least, and you think you've learned something important, chances are it was already done in Germany in the late 1800s. And that was the case here too. a German biochemist named Friedrich Miescher had discovered the chemical entity of DNA. It was a rather glamorous experiment. He was interested in analyzing pus from wounds, which were at the time known to be composed mainly of shrunken nuclei from white blood cells. These are dying white blood cells, and all that's left essentially after they're degraded is a nucleus filled with whatever is normally in the nucleus. He extracted that material in acid, and he found by a number of criteria it wasn't a protein, but a strange structure, a strange compound that had a lot of phosphate in it, and he called it nuclein because it came from the nucleus. But he described the isolation of DNA and noted that chromosomes are composed of both this nuclein, as he called it, and protein. Now, at this point in time, everyone knew that proteins were complex. Proteins do all the business of the cell. Proteins confer directly or indirectly almost all of our specific characteristics. Nucleic acids, as I'll tell you, are really simple. And so everyone assumed at the time that the genetic material had to be protein. It was the only moiety of sufficient complexity that one could imagine it doing all the different things that we know heredity can do. What began to change that notion, however, was another, what on the surface is a simple experiment done by Frederick Griffith in England in 1928. Now, this is the pre-antibiotic era, and one of the major causes of death in this time, and for many years thereafter, till the discovery of penicillin, was pneumonia. And a particularly virulent form of pneumonia was conferred by a microorganism, pneumococcus, which has been studied actually from the onset of the inception of this institute. Now, what was known is that there were at least two forms of pneumococcus with respect to their virulence. One form was sort of small, and if you can't see it in this picture, but was rough, had a rough surface, and this particular form of pneumococcus was completely non-pathogenic to a rabbit or other organism. So if you inject this rough pneumococcus into a rabbit, it happily bounces around. There's a second form called the smooth pneumococcus, and this form is virulent. You inject it into a bunny, and that bunny doesn't do so well. This is an artistic rendering of that. Now, what Griffith found was that, quite amazingly, that if you took some of this rough pneumococcus and mixed it in with a heat-killed version of this smooth pneumococcus and incubated it, you now would have an extract or a mixture that would kill a bunny. And if you go back and look, what you would find is that this rough pneumococcus had been converted into this smooth one. So something about the heat-killed extract from the smooth was able to transform the rough into a smooth, virulent bacteria. Well, it's not clear, to me at least, what Griffith made of this or what he did moving forward. His primary interest, at least when he was started, was to understand the pathogenesis of pneumococcus. And in fact, many vaccines were directed against the moieties that compose this smooth coat. But it was left to Avery, McCarty, and McLeod to try to purify whatever it was in this smooth-killed extract that was able to convert the rough to the smooth. And they embarked on this work. Avery had a prior interest, actually, in pneumococcus and pneumococcal vaccine. And as Paul mentioned to you, in 1944, they identified the material, the transforming factor, as DNA. And this is a picture of Avery, McLeod, and McCarty, published in 1944. With this publication, however, there was a major controversy that included many institutions around the world, not the least of which was Rockefeller. Because in addition to Hotchkiss, who was quite excited about this result and believed it deeply, others here, including Dr. Mirsky, disbelieved it, arguing that that DNA is a type of molecule that would seem to be too simple to account for all the variation seen within species. And that's because DNA indeed is simple. It's simply a repeating polymer of 4 units. Actually, much of what's known about this molecule early on was defined by a chemist here at Rockefeller named Phoebus Levine. And he identified the building blocks of DNA as being 4 simple moieties, 3 of which are shown here. They all contain a sugar in the middle, phosphate on one side, and another nitrogenous molecule called a base on the other. All 4 of these bases, 3 shown here, are the same except for the particular characteristics of this added nitrogenated molecule, which could be either called adenine, guanine, cytosine, or the 4th thymidine. And so the question is, how does a molecule with 4 building blocks create all the diversity of life and all the diversity of heredity and all the variability we see in each of this? And that was the question that Watson and Crick and others took on. How is it that the DNA works? And in 1953, they discovered the structure of DNA by identifying DNA's actual structure as the double helix. And I'll tell you how this works in a moment, but just simply say that they realized immediately that DNA could in fact serve as the blueprint for all of life, or all of the elements of life, both in the form of proteins, which then do much of the business or all the business of the cell. And so DNA, if you can think about it, is sort of the master plan for every organism. It's copied into a set of blueprints called RNA. It's sort of a transition molecule that provides the blueprints for this master plan. And this RNA, once it's made, encodes proteins. So the DNA is the master plan, as it were, for every organism. And the way it works is this. While the structure of DNA itself is too simple to account for all the variability in life, it's the sequence, Watson and Crick realized, that holds the key. And so each of these bases are shown here, and each letter refers to each of the 4 that I mentioned, cytosine, thymidine, adenosine, guanine, and they form a linear polymer. And actually what they realized is that you could have 2 polymers with anti-parallel sequences bound to each other. This sequence gets copied almost directly into the RNA, and the RNA in turn is read out into proteins by a genetic code. So the DNA linear sequence of bases is read out with a linear array of proteins. It goes something like this. Actually, Crick was the first to realize that the code was a triplet code in some absolutely elegant and beautiful experiments. So the DNA bases are read out 3 at a time. Each specific 3NA base pair sequence is shown later by a number of groups, reads out a different amino acid. So GCA means the amino acid alanine, AGA means arginine, and so on. And you go on in a linear array, and these bases encode, as it were, individual proteins. And the genetic code was solved in the early 1960s as a series of triplets. And so what you can see here is that each of these combinations of 3 correspond to different amino acids with some redundancy. UUU means Phi, U is the equivalent of T, I should say, ACA is threonine, and so on. So this is how DNA works. It encodes a series of units of linear bases that are read out into proteins, and it was shown previously that each of these proteins in turn is one of those hereditary elements. So a gene, which was an abstract principle for Mendel, becomes a concrete moiety in the form of a piece of DNA encoding for a particular protein. Now, this was more or less the background before the onset of the molecular biology revolution. But with the development of a whole set of methodologies, not the least of which was the ability to actually sequence this array of DNA, a project started in the late 1980s known as the Genome Project. And the objective when it started, and I should say that a major initiator of this was Jim Watson of the Watson and Crick discovery I mentioned a moment ago. Their objective was to sequence all the linear array of DNA in the human species, 3 billion bases. One linked to the other in a linear array on 46 chromosomes, or what composed the master plan for a human. From this, they could identify a gene list, all the different segments of DNA that encode for different proteins. And moreover, they could then begin to ask to what extent there's variation in these genes and how might they correlate with phenotypic differences among us. In 2001, this DNA sequence was made public. It was deposited in a database. I'll show you an example of that in a moment. There was one quite curious and interesting feature of this original sequence. It was always assumed that the complexity of humans would require that there'd be an enormous number of genes, more so than any other organism. Indeed, humans have fewer genes than the plants that Mendel studied. There are only about 22,000 genes, it's now estimated, in the human genome. So out of these 3 billion linear bases, it's parsed up into about 22,000 protein coding sequences, pieces of DNA that encode for amino acids composing proteins. And you can go to any computer, you could go tonight and search up, say, chromosome 11. And you can see in your computer a linear array of the chromosome, and underneath this would be each base, one after the other. Sort of like a tablet, as it were, that contains all of our blueprints. And shown below in these letters are each of the genes linearly arrayed along these chromosomes that make one one protein after another. Now, one way to think about these 22,000 genes would be to analogize them to the periodic tables. I think you probably all recall from chemistry that all molecules are composed of atoms, all molecules are composed of elements, and there are a finite number of these, and they're listed here. And so any chemical entity can be described based on its composition with respect to these building blocks. This genome project, in a sense, has provided to biologists a similar set of building blocks, albeit much larger for the human species. There are 22,000 building blocks. Now, herein lies a major difference, however, between the elements and the genes. Elements don't change. They're the same. They're used in different combinations in different molecules. But in living organisms, the nature of these elements can be variable. So for a given gene, a human might have one version, but the sequence might be different in a plant, so the resulting protein might be different. And it's that difference that confers the phenotypic or observable differences between species. Think of these base differences as a spelling difference. One organism might simply spell out, "I am the gene," another might spell out, "I am the gyne," and the result would be a different protein being synthesized. And these different proteins differ in their amino acids, and as a consequence, their protein. Now, at some level, this explains why children of humans don't look like giraffes. As you're probably aware, humans and giraffes are different. And if you weren't aware of this, then I'll call your attention to a recent news report.
A human-giraffe hybrid has been discovered in East Africa. Thought to result from a male human copulating with a female giraffe, the hybrid cannot speak, but is capable of performing simple mathematical calculation.
So giraffes are different than human. Giraffes don't speak. We speak. They have long necks. And so why don't you look like a giraffe, and how is it that humans can speak? Well, our sequences are different. So our repertoire of genes, while the building blocks are the same, the DNA sequence and protein sequence is different. Giraffe DNA and protein sequence resemble ours only to the extent of about 85 to 90, more than a chicken, but less than a chimpanzee, and less than Neanderthal, whose DNA are actually being sequenced as well. So it's this variability in the DNA sequence, in some cases relatively subtle, that confers phenotypic difference on species and allows this template, or the master plan provided by DNA, to lead different fertilized eggs to develop into different organisms. Now, while there's DNA variation between species that explains all the phenotypic differences that distinguish them, there are also phenotypic variation within a species. And it's these DNA base changes it's DNA base differences that are more subtle than this that account for those phenotypic differences. So as I mentioned, the human genome has 3 billion of these single units, these letters, as it were. And within the human species, on average, about 1 in 1,000 bases differ. So of our 3 billion differences, there would be about, of 3 billion bases, there would be about 3 million aggregate differences between any two individuals. And it's those differences and the spelling differences in their proteins that account for all the variability between them. Now, that then, in a sense, answers another question I posed, is how come I look like my parents? Well, it's because your parents pass on to you the same precise spelling variants that they themselves carry, and you in turn pass on those same specific spelling differences to your children. This is why different humans typically look different than other humans, unless, as I'll show you, they're identical twins. Now, there's a way to estimate the relative contribution of these specific variants to the differences that distinguish all of us. The classic way to estimate the contribution that these spelling differences confer is to systematically compare identical twins to fraternal twins. Identical twins share genes and environment, and at least by comparison to identical twins, fraternal twins only share their environment, and they share less of their genes than do identical twins. So if you systematically compare groups of identical twins to groups of fraternal twins, the relative similarity in identical twins in excess of fraternal twins gives you some insight into whether that trait is genetic. In a sense, you subtract out environment and ask what added similarity is the result of genes, or spelling differences, as it were. So this is an interesting family of identical twin brothers marrying identical twin sisters, and all their children are identical. because they passed on precisely the same DNA sequences from generation to generation. It must be an interesting set of relationships there, it occurs to me. Now, from estimates of this sort, you can conclude that genes, or more precisely these variation in genes, contribute to almost every trait that's been yet studied, with variable degrees. Now, my interest is in obesity, and there's a huge genetic contribution to obesity. 80 to 90% of the variability in obesity, in the incidence of obesity, can be ascribed to specific spelling differences that we're actively studying. But schizophrenia has genetic contributors, so too does diabetes, hypertension, alcoholism, epilepsy, heart disease, breast cancer, and so on. And it's quite possible today to actually track down the molecular basis of these differences. In the simplest form of analysis, what you simply do is take a family in which some individuals have the disease and other individuals don't. And now you ask, is there a spelling difference that is always found in the patients who have the disease and not in the people who don't have it? So in this example, the disease would be likely caused by this spelling difference from an A to a C. And this is always associated with disease. And by knowing that, we can actually understand what's different about the protein in this disease state. It was a variation of this sort of approach that allowed investigators to identify the cystic fibrosis gene, and that's shown here. There's a deletion in the cystic fibrosis gene that leads an amino acid to be missing, and that missing amino acid perturbs the function of that protein, and humans get cystic fibrosis. And single gene disorders have now been identified for muscular dystrophy, Huntington's disease, cystic fibrosis, hereditary breast and colon cancer, familial hypertension, diabetes, and so on. As Paul alluded to, my laboratory studies obesity. There's a single gene difference that leads this child to weigh 3 times more almost than a child without that mutation of a similar age. This child is missing the hormone leptin, and in our laboratory we're able to show, at least in animals, that this hormone can normalize the weight of this child. So there are single gene disorders that cause obesity. There are single gene disorders that cause sleep problems. Narcolepsy can be the result of a single gene disorder in a gene called hypocretin. There's also a gene that confers differences in speech. In fact, this gene is unique in humans. There's a unique sequence in human as compared to every other species. In this family, a mutation actually leads the affected children to not be able to speak. And interestingly, all other mammalian organisms have the same variant as these children who can't speak have. And so it appears during human evolution, a base change in this gene identified by Tony Monaco and colleagues in the so-called FOXP2 gene has conferred the ability to speak, and may actually have something to do with why it is that humans can speak and giraffes, for example, cannot. Now, thousands of single gene disorders have been identified. A few are listed here, and there are genetic tests for many of these. Genes that cause cancer, cystic fibrosis, coagulation disorders, adverse drug reactions, and so on. Now, there's a continuum of genetic disorders that extend it from one extreme, as these single disorders, with an almost purely genetic contribution, to a whole host of other genes that are a combination of genetic and environmental contributors. There are actually ways to identify these more complex genetic factors as well. And the basic way this is done is illustrated on this slide. Imagine you had a DNA sequence difference that was always present in the affected individuals for a particular disease and never in the unaffected individuals. And all the other DNA sequence differences that you could test by DNA sequencing were at equal percentages in both groups in this case. Well, what you might infer, based on if you had particularly robust statistical arguments to back it up, is that that DNA sequence variant, which is designated orange here, basically contributes to affected status for that particular disease. And this is being done systematically now for tens of thousands of genes in tens of thousands of patients. And from this, ever increasing numbers of genes that contribute to disease are being identified. And as I began by telling you, the availability of a complete genome sequence from anyone who's willing to pay for it at ever decreasing prices is going to change our understanding in time of the genes that lead to disease and the specific spelling differences that confer disease susceptibility. Now, at the moment, the the number of individuals who've availed themselves of this service is rather small. I don't know the exact number, although none of these companies appear to be doing especially well. But it's everyone's expectation that in time, one's DNA sequence will be available for amounts as low as $1,000, amounts that many people in America could afford, at least until recently. And when that happens, when we all can have our DNA sequenced, and we can all have correlated for us our particular DNA variants with our disease susceptibility and other traits, then things will change for all of us. And this is something that one can expect certainly in our children's lifetime. In fact, at that point, the world could look something like in the movie Gattaca. Gattaca actually stands, it's all GA's, T's, and C's, you'll note, and it refers to a world in which this information is available and is frankly misused in important ways. And I'll come back to this at the end. It's important to think about not only how this information can be used for good, but what some of the dangerous and potential issues are. In Gattaca, assumptions are made about people's potential simply based on their DNA sequence. And that's probably an overstatement for most things. For most human characteristics, it's a combination of genes and environment. So useful information is provided, but it is not, does not preordain all. Now, with this as our expectation moving forward, I want to make a few predictions, bearing in mind the quote from one of our great philosophers, that making predictions is risky, particularly about the future. But I will predict that the Genome Project, the genomics, will be at the center of 21st century medicine in the coming decades. As I mentioned, there are still millions of single base pair differences in our genome, and the clinical significance of many, perhaps all of these differences will be established, and then personalized medicine will become a reality. What does this mean? It will provide a better understanding of the molecular basis of disease. A better understanding of the molecular basis of disease will make it possible to identify people with predispositions before a disease develops, so preventive measures can be implemented. Medicines will be developed to target specific causes of disease rather than symptoms, and will moreover have the ability to tailor treatment to specific abnormalities in each patient. At the same time, as I alluded to, there'll be many medical, social, and ethical challenges, and the practice of medicine will become more complex, and this will also potentially worsen health disparities. Now, I want to give you a few examples of how this has or will change medicine. There are genetic variances that confer susceptibility to asthma. There are genetic differences that you could have tested for now that define differences in susceptibility to Alzheimer's disease. In some cases, which are rare, these will predispose to very early onset Alzheimer's, but in other cases, later onset Alzheimer's, but in a very sizable number of patients. Gene variation can also explain differences in response to drugs. Drug responsiveness or drug metabolism is genetically encoded, and there are a huge number of instances where wrong dosages injure people in serious ways. There's an example of this. A very serious childhood leukemia is known as ALL. The commonly used chemotherapeutic drug to treat this is metabolized by a gene called TPMT. This comes in two different versions. If you have the wrong version of this gene, if you're a child getting this medicine and you have the wrong version, you don't metabolize it properly, and what for everyone else is a normal dose becomes a lethal dose. And so testing for this variation is absolutely critical if you're going to contemplate using this drug. And of course, this is just one of an innumerable number of examples. For example, the response to statins, which many of you are on, I would believe, the response is highly variable depending on the variation in this gene, the CETP gene. This is a cholesterol metabolism gene. Now, this is all information that's going to become relevant, and moreover, when the DNA sequence for all of us is available, then sooner or later some or perhaps all of these connections will be made. So what are the challenges moving forward, both with respect to drug responsiveness known as pharmacogenetics and others? Well, the future looks something like this. The FDA already okayed an implantable information chip into people that would provide information about their allergies, prior treatments, and so on. In your child's lifetime, that chip will almost certainly have their DNA sequence, at least if there aren't ethical concerns that constrain this. That DNA sequence will be provided together with information about the relative risk that that child carries for any number of diseases. So it's imaginable to me that each of us at birth in the years, well, everyone who's about to be born, I'm guessing all of us have already been born, but that you'll get a relative risk profile, and you'll be able to say, geez, I'm at risk of asthma or COPD based on my genetic variation, and if I am, then it's probably an especially bad idea to smoke, not that it's ever a good idea. There are other diseases such as cancer for which heightened surveillance might be indicated, and so on. You may get a personalized formulary with which drugs you'll respond to and whether or not there are safety flags in response to any of them, or whether any known drugs might elicit a specific complication. And what's even more complex about this, as I alluded to before as well, is that you won't get any guarantees. You'll simply get information that says the likelihood of this eventuality is either increased or decreased. And this is going to require that people get used to a level of uncertainty and simply minimizing risks rather than demanding a fixed and varied outcome. Will each of us also be facing a set of questions about what we'd actually like to know? There are no perfect specimens. We all have glitches, as it were. So what would you do, or would you even want to know if your unborn child could inherit a treatable disease? This is already faced by many parents for a disorder known as phenylketonuria, which is treated. Would you want to know if you were likely to develop Huntington's disease, a movement disorder that's very severe? Would you want to know if you or your children had an increased risk of developing Alzheimer's disease or heart disease? Would you want to know if your child had an increased likelihood of becoming obese? Which would you want to know? Now, the conventional wisdom is that people shouldn't find out about those things for which they can do nothing. That the only information that's useful is actionable. But of course, that's someone else's assessment. These are the sorts of decisions that everyone's going to have to make for themselves, and that you'll make actively if, in fact, you decide to get your DNA tested, particularly at a point in time where all these associations are known. There's another question that comes up, and that is, what should anyone else be allowed to know? Is my DNA private? Now, there are numerous bills and there's a great deal of legislation that in fact ensures genetic information does remain private as it's generated, so that individuals won't be discriminated against based on their DNA sequence or denied insurance. But I want to close by pointing out an example where patient privacy could come into conflict with the public good, and these are also the kinds of issues that we're going to have to face collectively in the years to come. And this example refers to a disorder known as long QT syndrome. This is a rare hereditary disorder of the heart's electrical rhythm that occurs in otherwise normal or healthy people. People with this syndrome, which is inherited as a single gene and for which a diagnostic test is available, are susceptible to an abnormally rapid, lethal heart arrhythmia called torsade de pointe, and if you get this, people faint within seconds and often can die. 7 different genes have been identified thus far that cause this disease. They encode ion channel subunits that are made in the human heart, and the disease can be identified by genetic testing. Now, if someone had long QT, should this be private? So would an employer or the public have the right to know that someone had long QT syndrome if that person were a stockbroker? Absolutely. A CEO, well, it depends what industry. A bus driver, an airline pilot. Do we have the right to know whether or not someone carries a genetic disorder that would lead them to sudden death if they were flying the plane you were on? It could be even more complicated. How should it be handled if we found out that the sibling of an airline pilot had long QT, knowing that there was now a 50% risk of the pilot for having the same condition? Now, I don't have an answer for these questions. They're complex questions, but they're the sort of questions that we're going to have to face collectively as this information enters the mainstream. So I'm about to close, and I just want to summarize for you now what I think the promise, prospect, and issues surrounding this revolution carry with it. I think we have the potential to define the genetic and ultimately, with that, the environmental risk factors for all common diseases. This will allow people to develop personalized preventive medicine. Gene-based therapeutics for single gene and complex disorders will happen. And I don't mean to imply that this is in the next year or even 5, but I think incremental progress toward this will certainly occur over the course of the decades in our children's future. I think this is going to require that we educate health professionals and provide the tools. No one can keep all this information straight in their head. You may have already read about debates about the electronic medical record. Well, if we're going to enter an era in which this information is acted upon, then that's a sine qua non. We're going to have electronic means for recording and analyzing this information. This is going to create healthcare disparities, as people either can't get that information or don't have the resources to act on it. And there are privacy issues that, on the one hand, I think everyone would agree represent a source of an individual's legacy, as it were, and it's their own information. But I think it's a mistake to ignore some of the complexities that come along with that. These are some of the issues that are in my children's future. These are my children in their room with one of their cousins. This is part of their future, but there's more to their future than this. And so what I want to close with is the subtext I alluded to at the beginning. In addition to the importance of understanding genetics and genomics for all of our health care and our future, and I think potentially a better world for all of us, I think there are some other lessons I would like to have communicated. Well, firstly, science literacy is going to be increasingly important for navigating the world of tomorrow. It's the world of health care, but of course it's not restricted to health care. Scientific advances become most comprehensible if you can understand the historical antecedents. All of the advances I talked about and every advance I know about was put in a particular context. It was a moment in time where something that was known, there was a problem that bothered someone, and someone solved that problem. And so the best way to understand a system or progress is to understand the questions that drove that person to do a particular set of experiment. And then finally, for scientists as well as parents, there's always another question. So with that, I'll stop, and I'd be happy to take any questions you might have.
Yes. You put up a slide which showed that 80 to 88% of obesity was explained by genes. And yet I'm wondering why 50 years ago obesity was so rare, and yet and today it's 2/3 of the population in this country.
My view on that is as follows, and it has to do with the fact that with the way obesity is defined. So we define obesity by a fixed threshold, where if you exceed the threshold, you're called obese. If you don't exceed the threshold, you're not called obese. So over this interval where people say the obesity rate is doubled, what's essentially happened is a bell-shaped curve has shifted a little bit. And so the number of people who exceed that threshold has increased, but actually the net change in terms of weight is nowhere near as great as you would imagine hearing those statistics. So over these intervals, for example, average weight change has been about 10 pounds for the population. Now that tips a lot of people over the edge, but it doesn't, it's not, at least from a geneticist's perspective, a huge change in weight. And so what's referred to there with respect to hereditability is the fact that major differences in weight, say by 50 pounds or so on, is largely genetic factors. More subtle differences, such as the 10 or 15 pound weight changes that I'm referring to that become relevant for a population shift, are combinations of genes and environment. You don't look convinced. I get this all the time.
I wonder how many people in this room think that the average person is only 10 pounds heavier today than 50 years ago.
Well, you know, it's sort of like the Richard Pryor thing. Who are you going to believe, me or your lying eyes? You know, the point is that, you know, we all walk around, we have our anecdotal experiences. You have no idea how many times I get the question, well, have you ever been to Disney World? Well, actually, that's where that picture was taken of my children. The fact is, no one was looking at this 10 or 20 years ago they were now. So everyone's imagining there's this big change because no one was sensitized to it before. And so in a case like that, I think it's probably better to just rely on the actual numbers you can get from the CDC, and the ones I quoted were provided to me by some investigators there. But this is actually a much longer discussion. I'd be happy to give you some articles or tell you more about it later or in the reception.
Could you just talk briefly about set point and how that relates to the genetics?
Okay, so I think what we're really learning is my credibility for genetics versus body weight control, which is what the question has to do with. So I think there's very good reason to believe, based on a whole set of physiologic experiments, that body weight is regulated at a very tight or narrow range for each individual. And actually, there's good evolutionary reasons why that would be. In the eyes of evolution, it's a big mistake if you're too fat, and there's a big mistake if you're too thin. And so there are biological systems that sync to balance the relative risks of being at either extreme. And the underlying mechanism that accounts for this set point, as it were, or this ability to maintain weight within a range, is comprised of this hormone we identified named leptin and other elements. And so what we have actually is a hormonal loop comprised of a hormone made by fat that sends a signal to your brain about how much fat you carry, so that adjustments can be made if weight changes in either direction. And that, as I alluded to or mentioned, is I think a benefit in the eyes of evolution, because it protects you from wild weight fluctuations that would leave you poorly adapted for one environment or another.
Question here?
I'm going to go back to genetics. Is there an understanding for why genes mutate if it's not inherited from either parent?
So genes mutate by basically as a result, or the spelling changes occur over time and then they're propagated thereafter. They occur usually either because of just an error in the machine that copies the DNA from generation to generation, and the frequency with which this happens is increased in response to agents that induce DNA damage. Certain drugs can do it, toxins and so on, but it can happen spontaneously with no environmental contributor either.
And so does that explain your estimated, when you said like 88% chance of obesity is the rest based on just flaws in the system?
So when you say 85% of obesity is genetic, and in fairness to the question that was asked, this is really for extreme differences in weight, not more subtle ones. What that means is that in any given individual who is extremely overweight, 85% of the differences can be ascribed to an aggregate of genetic or DNA-based differences. The remaining 15% is probably due to environmental factors, the nature of which we don't actually understand.
Is there enough genetic research happening now? And if not, what's missing, and how do we encourage more young people to go into research?
Geez, I mean, there's a lot of it. There's been a lot of resources poured into this field since the 1980s. Is there enough? I don't think, honestly, this is a field that's particularly hamstrung for resources. In some ways, what really is needed is a technical breakthrough that really lowers the cost for generating a genome sequence. Right now, it's very expensive. What is it, $100,000 to maybe even $1 million? And so that's the bottleneck in my estimation right now, because your ability to get a really robust data set for large numbers of people is limited, and the methods that are being used now are just inadequate as compared to what a DNA sequence would look like. So then you ask, well, how do you encourage the next breakthrough that really advances this to a significantly different level? And that sort of spark of creativity is something that just happens in one person somewhere and is hard to predict and doesn't always respond to people throwing money at it. That said, there's a lot of money in the private sector for this. There are a lot of companies trying to do it. And right now we're in a phase of incremental improvements that have actually gotten us farther than we even expected. But how we get to that $1,000 genome, which is where everyone agrees the world of medicine really starts to change, is just not clear. Paul, do you have an answer for that?
I don't have an answer. I'd add one or two other things. I think another important issue is getting access to data from people. And this is a sort of sociological phenomenon. That is, whether people will be involved and engaged in sharing their DNA and sharing the information. And I think that will become quite an issue. I think the second point about encouraging people to do it, I think this has got, I will say one thing, we do have to have a respect for science. And, you know, when we have half the country believing in creationism, instead of, which is so anti-genetics, that's a serious issue for us. So I think we have an educational issue, and I think we have sociological issues to do with collecting data, in addition to the science. There's a question there in the middle. I don't know if Jeff agrees. Yeah.
No, yeah, I agree. Well, I'm not in favor of creationism.
Oh, what a shame. That would have been such an interesting discussion.
Without expressing opinion on creationism, given cell reproduction, it basically copies the DNA from our beginning embryonic cells throughout every cell in our body. If a particular disease is 80 or 90% genetic or shown to be so, what is the outlook for curing that type of disease? Is it something that is on the order of magnitude of a decade's horizon, and B, is it something that you would cure at the embryonic stage versus sometime later in life?
So I'm glad you asked that, because it picks up on some of the other questions, and it has to do with something I alluded to, which is this issue about getting comfortable with percentages and likelihoods, or a level of complexity that's sometimes hard to digest if you're not used to thinking about medicine in this way. So when we say there's a percentage contribution, it means that many things contribute, and that it's not as simple in all cases as one gene causing one disease. Now, the other question you're asking is, how does a knowledge about a DNA's base change lead to a new treatment, or better yet, a cure, which, as you're saying, would require that you correct the gene defect in every cell of your body, or at least in some cells of your body. Now, to some extent, that's a lot of why stem cells have generated a lot of excitement. They provide an opportunity to maybe repopulate different organs in one way or another with cells similar or identical to your own that don't have that defect. In most cases, however, a knowledge about the genetic basis of a disease doesn't necessarily give you a cure, but it gives you an entry point into a pathway where you can manipulate physiology in a beneficial way. And probably the best example of that I mentioned, these are statins, which are drugs that lower your cholesterol. None of them necessarily treat the underlying problem that leads you to have high cholesterol or heart disease. But nonetheless, this drug, by some other mechanism, allows you to lower cholesterol in a manner that ends up improving your health. And so I think for most things, that's going to be the preferred route of therapy, or the initial route of therapy, although the potential for stem cells, I think, to get closer to what you're suggesting, I think, is something that's generated a lot of excitement. So I just want to mention, no one probably picked this up because it was, but part of the reason I used the giraffe analogy was because creationists like to invoke giraffes as some indication that, there's a higher being, which I think is probably true, but not for the reasons they say.
Although I'm personally interested in neurofibromatosis, I'm going to use cystic fibrosis because you use that as a single gene disorder. I have a question. If you're able to manipulate, will there be something that will be able to change that gene in all people, or is cystic fibrosis, they're going to find it and it's going to work in 70% of the people and it won't work in the other 30%? Like, that's my question.
I'm not sure I quite...
I guess my question is I know that, for example, in breast cancer, tamoxifen works in some people and doesn't work in other people. And I'm wondering, in things such as cystic fibrosis, do you think that people are going to come up with something that are going to work in, for some people and not other people, either in NF or cystic fibrosis?
So cystic fibrosis is, to some extent, a simpler case, although in the end it may not be quite so simple, in that everyone who has cystic fibrosis has a defect in the same gene. And so probably most of the therapies that would at least be palliative, short of, say, a stem cell therapy where you can replace, lung tissue and correct the defects. So short of that, if you're talking about something that alters the physiology in some beneficial way by reducing secretions or whatever, I think most people would respond similarly. But for most what are called common disorders, many different genes, completely independent genes, can cause the same symptom set. So my research is in obesity, and many different genes can cause obesity. In fact, we know probably a half a dozen genes, separate genes, that when defective can cause people to become massively obese. And what I think in cases like that will emerge is that different subgroups of people who look the same actually have different diseases or subtly different diseases and will respond to different therapies, in different ways.
My question is, if genes can mutate, or if you can make changes to them, or if they can suffer trauma, does that mean that that gene would be changed in a person's DNA and would be inherited differently?
So actually, this is a whole other topic I didn't talk about. So DNA changes can occur in any cell in your body. If it happens in a germ cell, a cell that gives rise to a sperm or an egg, then it gets passed on from generation to generation thereafter. But you can also have DNA sequence changes in cells of your body that don't lead to the next generation, but nonetheless have catastrophic consequences. If, for example, that DNA-based change activates a gene that causes cancer or that puts you on the path towards cancer. And there's a whole other level of DNA sequencing and implications for healthcare I didn't talk about, where you can actually categorize what's happened to the DNA in individual cells. And here's yet another example where cancer, for example, can be the result of very disparate genetic differences. So what looks like the same cancer, like lymphoma, actually are different cancers because there are different genetic defects in different therapies. So I think this technology has broader implications than just looking at your healthcare record prospectively, but has important diagnostic importance. Right now, clearly for cancer, but this will extend to other disorders too. This is more your area. Do you want to comment on that, Paul? Or?
No.
Actually, my question follows on that. I'm interested in celiac disease, which often is not diagnosed for some long period of time. And in the context of somebody's lifetime, they develop other diseases. So it's really about the pattern of diseases that you might be able to identify with a genetic screen. And that, in fact, should lead us to be able to understand why people get other things in addition to that.
Absolutely. So a lot of, I don't actually know very much about the genetics of celiac disease other than it is genetic. It would certainly, as I understand it, be useful to have a test so that people could find out that they had celiac disease earlier, because I think a lot of suffering goes on in between the time people develop symptoms and develop, get put on the correct treatment. At the same time, if we knew what the genes were for these things and what tissues they're in and what they do in these different places, it potentially has a lot of explanatory power for why a lot of these diseases affect different organs. Remember, if there are only 22,000 genes, what you immediately come up with is that each of these genes is doing multiple things in different times and places. So the same gene defect could do one thing in the intestinal cells and yet another thing in another tissue. And in time, a lot of these connections, I think, will be made clear.
You spoke about the genetic component in, for example, 50-pound differences. Could you characterize to what extent genetics are involved in differences that are less severe than that?
The way I would phrase it is this. I think there's a bell-shaped curve more or less for weight. It's slightly skewed at the high end, but there's more room at the high end. But if we just start with the assumption that there's a bell-shaped curve, I think for this trait and probably every other, as a general principle, the further you go out from the mean, the more obvious the genetic contributions become. So I think as you go towards the average weight or the people who are just off average, what you're probably talking about are subtle genes that increase your predisposition and that in combination with an environmental difference, or in my case, a lack of discipline, might lead you, 10 pounds heavier than you ought to be. I think it's actually an ironic situation because the people who are stigmatized most are the people who weigh the most, and they're the people who get the most heat about it, or who are stigmatized the most, and they can do the least about it, whereas people like me, who are a little heavier than they might be, get a relative pass by comparison, when in fact I think I would have a lot more control over the outcome than they would.
We've had a real treat today. I mean, we have gone through 130 years of genetics. We did it in 45 minutes. You had a master telling you how to do it. You're all geneticists now. You all understand it much better. And we have Jeff to thank for that. Thank you very much, Jeff.